Welcome here, everyone. This is a webinar on the NCBI eUtilities, a programming interface for the Entrez system. What we're going to try and do over the next little bit less than an hour or so is give you a general introduction to this API, give you several examples of how they work, a few examples of how they work on the web, what the website can do that's equivalent to these APIs, and then what you would do with the actual API calls themselves. I do want to let you know that this webinar will be closed captioned. There's a link on the page here for information about our closed captioning, and you'll be able to find that on the sites when this is recorded. I'm going to go ahead and start by saying a few things just about our webinar program in general. You can find information about NCBI webinars at the URL at the top of the screen, www.ncbi, etc., education/webinars. We do about one of these a month. We'd welcome your feedback and interest, of course, in topics that we might want to present in the future. So go to that page as you would like and see what's going on there. You can also access all of the recordings of previous webinars from that page also. We will, during this recording, not be taking questions, but if you do have questions, there will be slides at the end of this webinar where you can contact us, and we'd be happy to respond to any questions about the utilities that you would happen to have. So what we're going to really talk about today are the following general topics. So we're going to talk a little bit about Entrez, what the utilities are, and what they do. We'll talk about some of the basic functions of the utilities, the ESearch and ESummary utilities, and then a discussion of EFetch, which is really responsible for providing full records. And then ELink, which allows you to make connections between records in different databases or records in the same database. And finally, we're going to get to the point where we can talk about pipelines. In other words, how do I build an application that can move data from one place to another and start with searches or start with uploads and end up with data that has been linked or filtered in a particular way that would meet my needs. And at the very end, we're going to talk about a new tool that we're excited about called Entrez Direct, which is basically a suite of programs that lets you bring the utilities to the Unix command line. So now you have all the power of Unix behind you, and you can incorporate these utility calls directly into either shell scripts or call them from other applications, and use all the other Unix command line tools to sort and manipulate the output as you'd like. So let's get started. We're going to basically start with a general overview of Entrez and the utilities. The Entrez system at NCBI now houses approximately 40 databases and well over 1 billion different records. And the records range from literature records in relatively well-known databases such as PubMed and PubMed Central, to health records such as records about genetic tests or interactions between genotypes and phenotypes, human variations in disease, to genomes, full genome sequences, epigenomic studies, nucleotide sequences, next generation sequences, etc., gene sequences, expression information from the GEO database, and protein sequences and information in three-dimensional structures, as well as the PubChem suite of resources about small molecules, chemicals, drugs, and their bioassays. So all of this information and all of these various databases are available to you through the eUtilities API. Now, the one thing you should know about the API in terms of how to work with it and think about it within the Entrez system is that each Entrez database represents each record by what we call a unique integer ID, or a UID. And each database has their own specific name for what that ID is. And some of them you may have heard of before. For example, PubMed uses what we call a PubMed ID or a PMID. PubMed Central uses the PMC ID. Protein and nucleotide both use an ID that's usually referred to as a GI number. But the point is that all of these IDs are integers. And that's the important point. They are not accession numbers, they are integers. And it's these integer IDs that the eUtilities use to reference individual records. The other part of this table that I'm showing to you is the name of each database that you need to provide to the eUtilities API. In general, it's pretty close to the name that you would think of, but just to be aware that there are particular database names that you need to use. And all of this is in the documentation that I'll show you in a few minutes. So the e-utilities themselves are a suite of FCGI programs that live on our servers that you then send requests to, and they're just standard URL requests. So the interface is a fairly stable interface. That's the one thing that we would really like to communicate to the community, is that if you had a URL for an e-utility call that was produced back in the 1990s, it very likely would work today. So the interfaces remain stable for a very long time, and the interface itself is, our commitment is to try and keep it to be stable as much as possible going forward. And so these individual e-utilities have a stable interface, a stable group of parameters that they take and do a particular function. And we'll talk about these as we go through: search, summary, fetch, link. So what do you need to use these utilities? Not a great deal, honestly. You need an internet connection, of course. The better the internet connection, the better your response will be, particularly if you're wanting to deal with a lot of data. It's good to have a fast connection. You could use these things in just a web browser. So if you just have a single call and you just want to get some information back quickly, you can use a web browser. But I suspect that most users would need to use a programming language so that they can then submit these requests automatically and then process and parse the information they return. And any language that can provide and receive an HTTP request, so they can post a request and then receive it, is perfectly suitable. So I've listed some examples here: Perl, Python, C, C#, etc. But any language that can handle HTTP requests is fine. So what do the utilities do primarily? Well, here are the basic functions. So search takes a text query and returns you that set of integer IDs, right, that you need to go further. All right, so those are the IDs that match that particular text query in the database of your choice. You can take that set of IDs and then provide those to ESummary, which will then give you what we call record summaries or docsums. So those are short XML blobs that give you a little bit of metadata about the record, but will not necessarily give you the full record contents, but it's enough to get a sense of what the record is. You can also take that same set of UIDs that return from ESearch and provide it to EFetch. And EFetch will provide you with the full data record in a wide variety of formats. For example, you can get PubMed abstracts or PubMed Medline format, or in sequences you could get FASTA or GenBank or XML formats back. If you have a set of UIDs in a particular database, for example, gene, and you wanted to find proteins, that's what ELink can do for you. So ELink will take a set of UIDs in a particular database and return you a set of UIDs related to those IDs in a different database. So you can give ELink gene IDs, and it can give back to you protein IDs, PubMed IDs, nucleotide IDs, etc., that are linked in some particular way to the input set of gene IDs. And finally, I'm not going to talk a lot about ePost, but Post lets you take a set of IDs and upload them to our server so that you can process them further. And there are some situations where that can be useful. We may talk briefly about that in a few minutes. So let's start with search and summary, the basic functions of what happens with the utilities and with Entrez. And what I'm going to do really to start with is get away from the PowerPoint and go to the web and do a couple of different examples here so that you can see a little bit about how these things work. So here's the NCBI homepage, standard web browser. What I'm going to do is navigate over here to PubMed and do a simple query: asthma children. So as you can see, we get results back. What I'd like to do is show you that you can limit results in particular ways. And already I've already limited the results over here to reviews and free full text. These are things that can be checked or unchecked at will. All right, so if I did not check them, I would get 42,000 back. But if I'm interested in reviews or interested in free full text, I can check those and then automatically get that limited set back. So now I have 800 records that are reviews and free full text, then also correspond to a query of asthma in children. So this is the kind of thing that people would do on the web. Now, if you're interested in doing this and using ESearch as an API call, the one thing you'll notice is that the review and the free full text limits are not in my query at all. So if you simply use asthma children, you're going to get back that 42,000 set of records. I don't want that. I'd like to get these back. So you need to be able to understand how you put additional terms in your query to execute those limits. If you scroll down on the right, there is a section called Search Details, which shows you exactly what Entrez has done with your query and shows you all of those particular terms. If you click See More here, it actually lets you see it a little bit more cleanly, so you can see the full query. And it also shows you down here translations that have happened between your input text terms and then the full query that Entrez actually executed for you. So you can see that asthma and children were both translated into several different mesh terms, and you can see that up here. And you can also see that there's a term here, review, PTIP, or publication type, which might be nonsense to you, but this is a field in Entrez, and now Entrez is just showing you what it did. And this thing, LO ATRA free full text, again, you can see down here is the translation of the free full text limit, all right, into a query language that Entrez understands. So what you could then do by looking at this is take this term here, and if I was to put that up in my query, I'll put and up there and then do that, and then do the search again, right? What I will do is I'll retrieve that same set. And if I was to take the limits off, in this case, you might say, well, the limits are wrong. But if I take those limits off, you see that the set remains the same. So this is a way of understanding how I can get the right query to go back into the Entrez system as an API call. So this is the thing I would need to use as my full term for ESearch. Another example is if we go to the gene database, I can search for a particular gene, human FOXP2. And again, I will now see in search details a slightly different query than I put in. So human FOXP2 is fairly simple. Things have happened to my query. So now I see that there is an alive property term that's been added in to this particular gene query. Now, I may not have expected that, but if you notice on the web, there's a current only filter that's automatically been applied for you. So to make that actually happen in an API call, you would need to add this alive property. So the point is, use search details to really understand the way that Entrez is processing a query, and then that will greatly help you understand what's going on and getting the right call for ESearch. Now, the things you're seeing here on your results is effectively a call to ESummary. All right, so really on the web, what happens is you do an ESearch call, and it's followed immediately by an ESummary call. So we can see those PMIDs here. Here they are. Those are the IDs returned by ESearch. But then on the web, there is an immediate ESummary call that then goes and gets these docsums for you. So, and that also happened in our gene example. So these are also records that are taken from the docsums from the gene database. So that's essentially what happens, all right, from search and summary. Now, what I'd like to do is return to the PowerPoint, just remind you again of what we just were talking about, that what happens when search and summary and a PubMed search is that they happen together on the web, and the same thing happens with gene. So again, just to remind you, we need to think about all of those filtered facets, other kinds of limits that are present in the PubMed or Gene live search that you can then add to your API term. So when you create the eSearch term, what do you need to do? There's a base URL, which is essentially just eutils.ncbi.nlm.nih.gov. It's right here, /entrez/eutils. That is a standard URL that is always used for the eutilities. It's a prefix. And then after that, esearch.fcgi, and then you give it parameters. And it's fairly simple. The parameters for search is one, a database, DB equals PubMed in this case, and a term, which is the term. So we used asthma children, and I've replaced the space with a plus. So in the APIs for Entrez, if you just replace spaces with pluses, it's fine. It will then do the correct thing. But again, the facets, those review limits and the free full text limits are not directly accessible here. You have to add those in. So I've done that down here for you. And again, this looks a little bit more complex, perhaps, because I've URL encoded the square brackets as the %5b, and these are standard encodings that you can find on the web, and %5d for the closed square bracket. So you can see that those have been put in. But basically, these are the things we got back from the search details on the web. So now we have the correct call. When you actually execute the call, the ESearch call, what you get back is an XML document that has several interesting things in it. One of them is it has a total number of records found. Here we go. Now this is 810. I did this a few days ago, so now it's up to 814, but it gives you the count of records. It also gives you the total number of records that it's showing you in the XML. Notice that it didn't give me all 810 IDs. It gave me only 20. That's the default setting. If you say ret max is more than 20, then it will give you more IDs. And ret start is, again, the identifier of the initial element in the return set. So 0 is the first element. So I could increase this element if I wanted to look further down into the result set and get a different set of IDs back. Also, the number of matching IDs here, again, is ret max. So if you set ret max to 20, you'll get 20 IDs returned in the XML. All right, so ESummary. What does an ESummary call look like? It's pretty simple. So again, the base URL, esummary.fcgi, and then you give it the same kind of parameters, db equals PubMed. But there's a difference here. Now we give it ID. So ID corresponds to the unique integer identifiers, all right, for that particular database, in this case, PMIDs. So in this case, I've just chosen 2 PMIDs to use as an example, and it will give me those 2 doc sums back. Now, just a couple of little tricks to note. You can get up to 10,000 records back in one eSummary request. So if you need to get more of them, you can loop, you can do more things, and we'll show you a few tricks about that in a couple of minutes. And again, this ID must be the unique integer IDs, all right, for that particular record. You cannot use accessions. So particularly for those of you who work with sequence databases and are used with GenBank accessions or reference sequence accessions or other kinds of accessions like that, you cannot use them in eSummary. You need to use the unique integer IDs. So the GI numbers, for example, for nucleotide and protein. Here's the URL. It's simple enough, right? You post that, you'll get the doc sums back. Now, one thing that we should be aware of, and that might save you a lot of time, is that you can short circuit, in a way, this movement of ID lists between the utilities by using something that we call the Entrez history. Right? So what this lets you do is represent an entire set of IDs with only 2 parameters. So the way it works is, in the ESearch call, what you do is simply add another parameter that says use history equals Y. If you do that, it then tells ESearch to post the results of the search onto the Entrez history server, and what you will see in the return is that you get 2 different parameters back that you didn't see before. One of them is called a query key, which is an integer that basically tells you what data set you're looking at, so that query key can increment, all right, and each set will be unique. But there's also a web environment string. This is a lot of gobbledygook, but don't worry about it. You just copy it and use it. It essentially is a cookie string that tells Entrez the address, all right, of your various query keys. So you need both. You need a web environment to show Entrez where to look, and then you need the label of that particular data set that's at your web environment. So if you take these 2 parameters, the query key and the web environment, and provide them to eSummary, it effectively replaces the ID parameter. So you don't need the ID parameter anymore. You give it the query key and the web environment, and it puts the entire set onto eSummary. So this is a very efficient way of doing a communication between search and summary when you need to do something like that. So let's see how this works. So here's a few URLs. What I'm going to do now is switch over to a text editor that I have over here with some of these URLs in it, and let's just take a look at how these work. So this is the original PubMed search. So I'm going to go back to a browser, and again, any of these utilities, you can just paste the call into a browser and see the return. Right, so now you see the live return is now back up to 814. Here's 20 records, and so this is what we get from eSearch. right? And also, I'm not going to talk a lot about this, but you'll see that in the bottom of the ESearch call, you get the full translation stack. So this is how Entrez processed your entire query. This can be useful. I'm not going to spend much time talking about it, but just so that you're aware that it's there. The ESummary call. Here it is. So if we copy this, put it up in our browser, what we get back are doc sums. All right, so this is an XML document. You'll see here that there's a document set. All right, it's one document set. Here's a document summary. Here's a second document summary for those two records that I provided. Each document summary has a variety of information in it: the date, the publication, the author list, the title. There's information about its publication type, DOI identifiers, etc., etc. So it's a fairly good document. There's a lot of information in ESummary, and so I'd always encourage people to look at the ESummary results, because many times that's all you need. And it's a little bit more efficient than using EFetch in general. So if the summary works for you, then by all means use it. Now, if we have the history, right, applied, here we go. So I'm going to just show you what happens when you use history equals y on your ESearch call, and you'll see these two parameters have now appeared. All right, so here's the query key. Everything else is the same. Again, it's 814 results. Here's my 20 IDs, are the same IDs. All the rest of this is identical to what we saw before. But now we have a query key of 1, and we have this bizarre NCBI web environment string. So that is the thing that you would then need to put into an ESummary call to then take this entire set of 814 records and download it. Okay, we've talked about ESearch, we've talked about ESummary. Let's now move on to EFetch, which gives you full records. So remember where we were? We were back here, right, in PubMed. I've got my query here. I've done my query. I'm all excited. Now I want to get particular records. So what I can do on the web is really simple, right? I just click on the title. That's it. That's my abstract. I just did an EFetch call. All right, so now Fetch went and got the entire abstract back, and now I can view that. Same thing would happen in Gene. Okay, there's the human FOXP2 gene. I click on its docsum title there, and then what's returned for me is a full record, right? And the gene record is quite big, all right, for something like FOXP2. It's several pages long, and that's what EFetch does, all right? So EFetch goes into our databases and pulls out those entire records. So I'm going to switch back to the PowerPoint now, and we'll talk a little bit more about EFetch. So again, the web equivalent is just clicking on a result. All right, so you click on an item in your result, you get the record. That's what EFetch does. Now, how do you do EFetch as an API call? Well, it's pretty similar to summary. Quite honestly, you say efetch.fcgi, and then you give it the database, PubMed, db equals PubMed, and again, you give it IDs. All right, so it's exactly the same syntax as a summary call, except that now you have options. All right, because Fetch gives you the full record, and it can give you that record in a wide variety of formats. So it's not necessarily true that Fetch knows what you want. You have to tell it. And so in our documentation, there's a table of all the different kinds of records that Fetch will provide for you. So for PubMed, for example, you can get ASN1, XML, Medline, a list of the PMIDs, which you might think is redundant, but in some cases that's useful, or the abstract. And you can get these things in a variety of formats as well. And so the formats that are listed here can be specified with two parameters: one, ret type or return type, and return mode or ret mode. And you'll see that what you need to do is use both of them. So if you don't use ret type and you don't use ret mode at all, the default will give you ASN1. If I want to get an abstract back, I need to say ret type equals abstract and ret mode equals text, and that's what I've done here. So all you simply do is add those parameters to your EFetch call, and you're in business. Now, the one thing that's important for everyone to understand is that EFetch does not currently support every Entrez database. This is a work in progress. So we do support many of the major databases, and this is often based on community demand. So many of our major databases are well supported. For example, PubMed, PMC, Mesh, and ELink Catalog in the literature area. In the sequence databases, the major ones, all the nucleotide and protein sequence databases, as well as PopSet, SRA, and even taxonomy are covered. And we cover gene, homology, SNP, etc. So there's a variety of databases, and we're adding more from time to time that Fetch will support. I believe we actually have support for ClinVar now, and it's not actually listed on this slide, so we need to update that. But we're continuing to get more and more of these databases online. But just be aware that EFetch support is not universal. However, ESummary is. So ESummary supports everything. All right, so if you can't get something to work in EFetch, you can always get it in ESummary. And so that's the thing to remember. Summary works for everything. EFetch will work for a limited set of databases. So what about this history thing that we were talking about? Does that work for Fetch? Sure, it works the same way. So if I say use history equals y in my search call, then I will get, again, the query key and web environment. I give those things to EFetch, and it will transfer the entire set to EFetch, and I'll get them. So it works exactly the same as it does in ESummary. One little point I'll make here, sometimes this is confusing to people, is that the use history equals y parameter is only a parameter for ESearch. You don't have to do that for ESummary or EFetch. ESummary and Fetch are already available to use the history at any time. It's only ESearch where you need to actually tell it to switch to a history mode. Now, let's say that you have a very large set of records that you want to download, like several tens of thousands, hundreds of thousands, or even more records that you want to download from a particular database. You can only get 10,000 at once, all right, from EFetch, just like ESummary. How do you do it? Well, you do a loop. And you use these two parameters, retStart and retMax, that I discussed a few minutes ago, because if you say retMax and you set it to some value of a batch, maybe 500, maybe 1,000, and then you loop over retStart, effectively you will download the set in batches of size set by retMax. So in this case, if retMax is set to 500, then I'll start retStart at 0, and then it will be 500, 1,000, 1,500, 2,000, etc. And this is, of course, very straightforward to do in a for loop or any other kind of loop that you'd like to create, and you can get the whole thing down. So this is a lot more efficient, all right, than doing 10,000 individual calls for 10,000 records. Instead, you'll only do maybe 20 calls or 50 calls or something like that and get a large set of data down. And so this is a really good approach if you have a large sample of data that you need to get. What I want to do at this point is just emphasize that many of the things that we've talked about are available to you in the documentation. So we've talked about doing those EFetch downloads, and there's an example with actually working Perl code in the EFetch documentation that lets you do that. And so I'd like to take just a brief diversion here, go over to the eUtilities help, which is simply eutils.ncbi.nlm.nih.gov. If you go there, you'll find this. And this is a book. It's an online book that has extensive documentation about the eUtilities. And most of everything that we've been talking about so far is presented here. So there's a quick start that will give you information, basic information about the eUtilities. Exactly the same kind of things that we've been talking about, basic searching, the kinds of URLs that you would need to create for searching, some sample output from eSearch, what kinds of things you can do with ePost, which we haven't talked about much. Here's the ESummary, etc. So there's a variety of things that you can learn here very quickly, all right, just by taking a look at this book. I also wanted to show you there are usage guidelines here about how to use the utilities. I'll talk a little bit more about these at the end. There are sample applications. In particular, for example, application 3, which is what we just talked about, retrieving large data sets. So if I wanted to get all, for example, here, the chimpanzee mRNA sequences, that's more than 50,000 sequences. I have a query, and I have a little Perl script here that will do the loop. So this basically retrieves data in batches of 500 using the same kind of approach we just talked about, like building those URLs and incrementing retStart. So there's a variety of information here. I really encourage you to explore this because there's good examples, there's good advice in here about things that you'd want to be able to do. All right. So what I'm going to go and do now is talk a little bit about another eUtility, ELink. So ELink is a particularly powerful eUtility in that it lets you get from one place to another. All right. And many of us do this all the time on the web, but we may not realize that we're doing it. For example, up here in this PubMed article, there's a link to full text. Now, if I click that link, what happens is I go to the publisher site and I get this particular document. If I have a subscription and if my institution has a subscription, those kinds of things are possible little barriers to you. But if you have a subscription or if it's free, you'll be able to just traverse that link. Now, that was an ELink call. All right, so even though this is beyond NCBI, this is an external publisher, ELink was something that allowed me to do that. Here's my gene record. Let's say that what I want to do is download the reference sequence proteins, right? The curated, annotated proteins that are products of this particular gene. So one way to do that, particularly in the API world, is to notice that there is a refseq protein link. So all of these things in this list are links that are accessible by ELink. So just by clicking on that link, what will happen is I will get a set here of 8 reference sequence proteins that correspond to all the different isoforms of FOXP2. So it's nice. And then I can download them from here. So ELink lets you do this. It lets you traverse between different databases at NCBI. So let's talk a little bit about how ELink works and how you can make those kinds of connections. So again, on the web, there are a variety of equivalents, right, for ELink. Clicking to full text from a PubMed abstract, clicking on a link that takes you from a gene to its refseq proteins. There are hundreds of these links in the Entrez system, and they are all described to you in the ELink documentation. There's a table linked there in the documentation that will show you all those different kinds of links. Now, how does ELink work? ELink is by far the most complex utility because it has a lot of different kinds of work that it does and because of all the different kinds of links that it can traverse. So you need to provide it not just one database, but now 2 databases. The database you're coming from, right? So that might be PubMed if I'm going to an external publisher, or to PMC. It might be gene if I'm going from gene to protein. So that's the DB from parameter. There's also a DB parameter, which is the database that you're going to, the destination or target database. So this could be protein in my gene to protein example. You then provided, again, a list of IDs. So this is just like summary and fetch, and then it needs the set of IDs in which database? The DB from database, right? Because that's where you're starting, right? So you give it the set of IDs in the beginning database, the DB from database, and then you get IDs back from the target database, DB. Now, CMD, or command mode, is an interesting topic in ELink that we don't have time to explore in full today. There are several different command modes in ELink. The default one is called neighbor, and this is all described again in the documentation. I'll talk about a couple of these command modes just so you get a sense of what ELink can do for you. So here's a particularly simple URL. Now I'm linking here from protein to PubMed for this ID. This ID is what kind of ID? A protein ID, because it's DB from protein to PubMed. Now, within Entrez, each particular kind of link has a name. All right, and that name is called a link name. And there are, again, hundreds of these guys. So how do you even keep this straight? How do I figure any of this out? Well, the good news is that there's another little e-utility called einfo that is designed to provide you this kind of information. So if I simply write a URL to einfo and give it database equals protein, it will list for me all of the links with some description available to me from the protein database. In other words, with DB from equals protein. So that's where you can go. Again, there's documentation on the ELink section of the book that will tell you more about this. But for example, a gene protein link name means a link that goes from gene to protein. But there's more interesting ones like gene protein refseq. That's the one we just did, right? It gives me not just any protein that's associated with that gene, but only reference sequence. In other words, curated proteins, all right, that are associated with that gene. I could have protein nuke core. But let's say I only want messenger RNAs that are linked to a particular protein. I don't want its genomic sequences or other kinds of sequences. Protein, nuc core, mRNA gives me only messenger RNAs linked to a particular protein. These are just a small number, many, many examples, all right, of links that are available to you. And a lot of these are underutilized. So I would encourage anyone interested in traversing NCBI data to explore these because they're well worth your time. Protein identical gives me all, again, the identical proteins to a particular protein of interest. Now, what happens? What does ELink give you back? Well, it gives you back an XML document showing you what all those links are. So what happens is the protein is the database we're coming from. Here is the ID that was given to us from protein, and then we're going to PubMed. Protein PubMed is the link name. That's the ID in PubMed. So this is a link from that ID to this ID in PubMed. Here's, again, protein PubMed RefSeq. So these are particular records that have been curated by the RefSeq group. So there's 2 of these. All right, so this is a link now to these 2 PMIDs. Now, you can imagine that in this case, you could have thousands of IDs that might be linked to something. Like if you link from protein to SNP, there could be all kinds of variations in a protein. So this could be a very long document. An advantage is another command mode of ELink called neighbor_history. And you might even now guess what it does. It posts the results of your link operation on the history server, just like ESearch does with useHistory=y, and now it only returns to you a query key and a web environment, right, for each link operation. So now for each link name, you have a separate query key. Here's 9 and 8, but one common web environment. So now you can take that query key and web environment and pass them to EFetch, pass them to ESummary to download either the docsums or full records. All right, so now you can begin to see how you can make these chains of events. So again, that's one link set. Here's the second link set. So again, you can explore that XML and parse that out. Now, before I leave the topic of ELink, I want to just, again, make a slight digression here because this is a particularly useful command mode of ELink for many of you who are interested in literature. Right, so what ELink provides for you is that path we did from PubMed abstract to that external publisher. All right, so there is a link command mode called prlinks. All right, prlinks, and if you give also ret mode equals ref, it's a little bit of an unusual syntax and it's quite unique for this command mode. It will not only provide you the link, but actually post that URL and take the user, right, to that particular record. So let's see a little bit about how this does and how this works. So here's my URL here. And again, this document is going to be available for you. It's part of the materials for the course. So you can come and practice these URLs as you'd like to. If I was to load that URL, and let me just go back so you can see it again, db_from is PubMed. There's the PMID. The command mode is prlinks. Ret mode equals ref. If I post this URL, what happens is, it's not an NCBI document at all. Boom, I go directly to the publisher. All right, so this is a really handy thing to use if you're designing a web page or a website, for example, where you'd like to provide direct links, all right, to publishers from PubMed abstracts. So I just wanted to let you know that particular trick because it's quite useful for anyone interested in literature. All right, so now let's get back and sum up kind of where we've been so far. So this is kind of the big picture of the way that utilities work, if you will. You can start in a couple of different ways, either with a text Entrez query, which you'd provide to ESearch. And I know we haven't talked a lot about this, but you can also start with a UID list and use ePost. In either case, you can get those IDs onto the history server, right? Either by a text query and then search provides the IDs to the history server, or ePost will provide the IDs directly. Once they're on the history server, represented by a web environment and query key, you can get the docsums, you can get full records, or you can link. Link can put those records of the output, right, in a different database, right back on the history server, where you can then get the full records or get the docsums. So this is basically a point where you can see that you can develop flows. and pipelines. I can start in one particular area of Entrez and move around the system and ultimately get the data that I want. So for example, I could start with a text query in gene, which might be a set of genes on a particular chromosome, or a set of genes in a particular region, or a set of gene symbols. And I would get a set of gene IDs, all right, that correspond to that search term. That set of gene IDs I could then put into ELink and get proteins out. Right now I have protein GI numbers. I could take those protein GI numbers, give them to EFetch, and get out some kind of protein record. For example, FASTA records, or GenPep records, or INSD XML records. So there's a variety of different things I could do. Right? So I hope this just starts your imagination ticking a little bit. All right, so you can think about a variety of different approaches you could do to actually build useful applications. How do you do something like this, all right, in the utilities? Well, you have to create a set of calls. So here's the first call, eSearch in gene with a particular set, all right, a particular term, use history equals y. I would then have to parse the XML output of that, take my web environment and query key, and put that into an ELink call. All right, I haven't done that. I've just represented them here as key in WebEnv, but that's what I would have to do. That ELink call will then give me, all right, either IDs or web environment query keys that I would then have to take and assemble an EFetch call, all right, ret type equals FASTA and mode equals text, for example, all right, and then provide the key in web environment. So that would be a way that I could do it. But you see that I have to parse, all right, the XML output of each one of these, right, before I put it into the next call. What I'd like to show you now is an easier way of doing this. And this is something that we call Entrez Direct. And what Entrez Direct is a way of using the utilities on the Unix command line, and it does a lot of the bookkeeping for you. Right? It does a lot of that parsing for you so that you don't have to worry about it so much. You essentially create a bunch of piped commands. So I may not even have to explain this, too much, because any of you who have used Unix can probably almost read what's going on here. I will talk about it, just for those of you who may not be so familiar with Unix. But basically what we have is a Unix command called eSearch. You give it -db gene, -query, and here's my query. Pipe. Right, so this symbol in Unix means I'm going to take the output of this command and send it directly to the next command. What this does is just say that I'm going to continue this command on another line. All right, so you can ignore that. Now I go ELink. It's another Unix command. Target is protein, so this is effectively the DB or destination DB. Name, this is the link name, gene, protein, refseq. Right? So notice I haven't had to tell it where the input's coming from. The input is coming automatically from here. So all the parsing is done for me. Pipe to EFetch -format FASTA. I'm done. So that does that whole pipeline. It starts in gene, it goes from gene to protein, and it then returns me FASTA. Now, Entrez Direct is something that you can get directly from our FTP site. Here's the URL. And I really encourage you to play around with this. It's a fun little tool. What is it? It's actually a set of Perl scripts. All right, so they're all little executable Perl scripts, but they're designed to work as Unix executable. So they have those command line switches, right, that you saw, -db, -format, things like that. Each script provides XML that can be interpreted by any other of those particular Entrez Direct scripts. So you can just pipe them together. What you need, okay, you need a Unix operating system. So that's either directly Unix or Linux, or Mac OS X, works just fine. You'll need Perl, all right, because they are Perl scripts. You'll need the standard library LWP simple for doing the URL calls, the HTTP requests. Almost every Perl installation that we're aware of at least comes with LWP simple. So we hope that this is not a very big barrier for most of you, but let us know if you have issues. The package contains a variety of things. It contains eSearch, Summary, Fetch, Link, and Post, and one called eFilter, which is not actually an eUtility, but is designed to perform an eSearch after a link operation. So for example, I wanted to link from protein to nucleotide, but I only want to get mouse nucleotide records. So I could filter the result set of my link operation for only mouse records, or any other thing you can think of. So filter is essentially ESearch, but designed to work after a previous one. So that's a fun little one. Another very interesting one and powerful one is something called xtract. And this is a new feature that is not part of the eUtilities API per se, but is part of the Entrez Direct suite. And it is basically an XML parser that lets you take out pieces of records. And it's an amazingly wonderful thing. And so that's a real advantage to using this. So here's something that xtract can do for you. It's a very simple case. It's a very powerful thing. And again, it's well documented in Chapter 6 of the eUtilities documentation. So I'd encourage you to go to Chapter 6 and take a look there and to see what's going on. Here's a doc sum from protein. All right, so the document summary, there's the protein ID, the caption is the accession, here's the title, and various other information, and the sequence link. Let's say, for example, what I wanted was a table of the accession, the title, and the sequence link. Well, what I can do is after my pipeline here, so ESearch, pipe to ELink, so gene goes to protein, and then to ESummary. Right, so now instead of EFetch FASTA, I'm just going to do ESummary. Then I pipe that output to xtract. Pattern is document summary, and then that's the actual element that it's going to use to loop over, if you will, or to explore within. I want these elements: caption, title, and sln. Right, and I can do it in any order I want, and it will output a table. It's a really great thing. So that's a quick example of some things that you can do. What I'm going to do now is actually show you live a few little scripts from Entrez Direct, or eDirect as we call it for short, that will let you do a few things just to get you started. So one is gene protein to FASTA. All right. The next one is producing a table of links from multiple genes. So this is a very useful thing to do. If you have a lot of gene IDs, and what you want to do is get particular protein records or any other kind of record, SNP, PubMed, whatever, for each gene as separate lists, this is a really great way to do that, because the standard link operations don't do that. They just give you one big set of output IDs, as opposed to an individual set for each input record. So this is kind of doing one-to-one links. It's very powerful, and this is an example of how you can do this very easily using eDirect. Another example is going from gene to docsums into a table, like what we just talked about. In fact, it is that very example, and then I can sort that table also. So let's take a look at how all this works. So back in this document, again, which is something that you'll have available in the materials, here are the 4 examples, and these are the actual scripts. All right, so you can just copy them and run them. So ESearch gene, here's my query, to ELink, all right, to protein, and getting FASTA. All right, here's the second one. Here's how I get those links. All right, so I'm going to search in gene. I'm going to pipe that to ELink, which takes me to protein, particular name, command neighbor, and then to xtract. And again, the documentation will show you more about how all this works, but there's a link set within that XML. I can loop over a block called ID list and extract out an element called ID, and then extract out of another block another element ID. So these are the input IDs in gene. These are the output IDs in protein. And you'll see that we can get those all put on one line. And then the next 2 examples are, again, the example pipeline I just showed you about going from gene to protein and then getting the table of accession, sequence length, and title. So you already have seen this, and all I'm showing you here in the 4th example is adding the power of Unix to this particular pipeline. I can use sort, right? And for those of you who understand sort, this might mean something to you, but what this means is I'm going to sort by the 2nd column. In other words, by sequence length. Right? So I'd like to sort the column that way. In other words, you can use sort, unique, grep, any of those things, all right, to process the output of these calls. So let's go. Here's the Unix command line. What I have here are the different shells, all right, shell scripts here that I've just described to you. So let's try the first one. Boom, FASTA. All right, so that was the one that went in from gene, linked to protein, and then did an EFetch and FASTA. You can see it's nice. I got a variety of those 8 fast days out. Done. What about the second one? This is the one that provides me those individual links for particular genes. So you can see what happens is it's not well labeled, but you understand from how you wrote the script what's happening. This is a gene ID. These are protein IDs that are associated with that gene. This is the next gene, 44967. These are the protein IDs associated with that gene, et cetera, et cetera. Very powerful. All right, this is a really wonderful function of the API that you can get these kinds of relationships very easily. All right, next one. Again, this is going from gene to protein to ESummary and extracting out the caption, the sequence length, and the title, and writing it out as a table. So I can extract any part of any record that I want, which is really cool, and then I can just put it in a table. So I can immediately get this into Excel. I can process this very easily. It's tab delimited. It can be delimited by any other character that I want. That's all part of extract. It's all in the documentation. So there's a variety of things that you can do. Now, the last example is just that, well, maybe I want to sort these by their sequence length because I really care about which ones are longest. Well, that's what that next script did. It just passes this to sort, and you can see that it's done that very nicely. So these are just a few very simple examples of some of the many, many things that you can do with Entrez Direct. We hope that you explore those and let us know if you have questions or thoughts or input about what kinds of things it could do better. All right, we're almost done. Only a couple of more things I want to talk to you about before we leave. One is that when you use the utilities, it's important for us to understand something about what you're doing to prevent problems. So we really encourage you to use these 2 parameters. These are non-functional parameters, but they essentially identify you to us. So in the case that there are issues, we can contact you about what's going on. So tool is meant to be the name of your software package. Now, if you're not developing a software package, you can just make up a name and give it to us. But it identifies all of your different queries and URL calls as coming from that same application. So if you're just writing a script, you can give the script a name, and that way we can associate those calls with that particular script. The email is meant to be a contact email so that if we detect problems, we can contact you. Now, here's the idea. If we detect a script that's a runaway script or a script that's giving us more than about 3 calls per second, which is our standard limit. So we try and advise you not to do more than 3 requests per second per IP. If you need to do more than that, we understand. You can contact us and let us know, and we can work with you about what would be a good time or way of you doing that. But if you're doing more than that, you could be blocked. All right, so because we have to let our resources be available to as many people as possible, if we're getting essentially a denial of service attack from a particular IP because of an enormous number of calls, we will block that IP. But if we have an email, right, if you provide an email, we will not block until we contact you. Right, we'll contact that email. If we can get a response back within a few hours, we won't block. So it's to your good to provide these to us just in case anything happens to go wrong. And it helps us help you. So that's something we'd really recommend that you do. Finally, I want to leave you with some places where you can get some more information. So again, if you're interested in these webinars, you can go to the education webinars page and find a lot of information about them. We hope you would do that. The eUtils, NCBI, NLM, NIH.gov is the full documentation. So of course you'd want to go there, use it as a reference. It's a rich source of material. And the Entrez Direct, I have to say, Jonathan Kahns, who is our developer of Entrez Direct, has done a magnificent job of providing many, many examples, including working code and shell scripts in there about ways you can do amazing things with Entrez Direct. And so I'd really encourage you to look at that. Utilities Announce is an email announce list that we provide to the community for announcements about the utilities. These are generally updates or new features and new functions or maybe new databases that are available now. You only will get about 1 message a month. You're not going to get spam from this, but it's a useful way to stay in the loop. And so we encourage you to sign up for that if you're at all interested. We also have a blog, the NCBI Insights blog. Here's its URL. And we do write about the API from time to time. And so we are going to be putting some of these examples about Entrez Direct up there so you'll have access to them. So I'd like to thank you again for your attention and your time in exploring these resources with us. Please let us know if you have additional questions. You can write to us at info@ncbi.nlm.nih.gov, and we'll be happy to respond to you. Thank you very much for your attention.